Let's return to today's list of themes again. In the last part, I will talk about nutritional signals during fasting. This chart shows the flow of the energy substrate in a human body during a 24-hour fasting. During a 24-hour fast, the liver releases 180 grams of glucose into the blood. These glucose outputs are derived from glycogenolysis and gluconeogenesis. 4/5 of them are used by the brain, and the rest is metabolized into lactate by blood cells and peripheral tissues. That lactate goes back to the liver and is resynthesized into glucose. So, lactate is one of the major substrates of gluconeogenesis. The most important source of gluconeogenesis is amino acid derived from protein in the skeletal muscle, and the rest is glycerol from triglycerides in the adipose tissue. Then, how is the flow of these energy substrates controlled? This list shows well-known fasting signals in our body, including hormones and metabolites. Looking at each organ of signal source, we can categorize them into pancreatic, intestinal, hepatic, adipose, and hypothalamus pituitary adrenal signals. Of these, the fat signal is the metabolite signal. Perhaps there might be unknown metabolite signals from other organs. This slide illustrates the content of the previous list. As the central organ of metabolism, the liver receives various information from each organ and regulates the metabolism. In the next section, I would like to describe two specific examples of new metabolite signals during fasting that have been discovered recently. The first topic is about alanine signal. Alanine signal was recently found as an example of fasting signal. Here, I would like to introduce the research results of Dr. Shimizu and others at the Institute of Medical Science, University of Tokyo. They found that alanine signal is a kind of fasting signal from a study of muscle-specific GR knockout mice. GR means glucocorticoid receptor. As shown here, muscle-specific GR knockout mice are resistant to proteolysis of skeletal muscle during fasting, so they are less likely to lose muscle during fasting. As a compensatory response to this muscle resistance, the adipose tissue is conversely smaller in knockout mice. The question is how this compensation occurs. As the answer to this question, they found that muscle-specific GR knockout mouse has a higher FGF21 expression level in the liver as well as in the plasma. FGF21 is a hepatokine, that is, a hormone secreted by the liver. Treatment of animals with FGF21 results in increased fat utilization and lipid excretion. Moreover, they found that muscle-specific GR knockout mouse has less alanine in the liver, which they predicted would trigger this compensatory response. In fact, alanine supplementation lowered the elevated FGF21 expression in the liver of knockout mouse. It also normalized the plasma FGF21 levels. In summary, fasting causes glucocorticoid-induced muscle proteolysis to provide the liver with the substrates for gluconeogenesis. In case this response to fasting does not occur in GR knockout mouse, The shortage of glucogenic amino acid alanine triggers the expression of a hepatokine FGF21, which in turn promotes lipolysis in the adipose tissue to supply glycerol as a compensatory substrate for hepatic gluconeogenesis. In this regulatory system, alanine acts as a metabolite signal to control the FGF21 expression, but the sensing mechanism is currently unknown. The second topic is about glycogen signal. This topic is based on our own research, published in Nature Communications in 2013. In this paper, we identified glycogen shortage as a new fasting signal in the liver during fasting. During fasting, animals maintain their energy balance by shifting their energy source from carbohydrates to triglycerides. In this work, we showed that liver glycogen shortage activates a liver-brain adipose neural axis, which has an important role in switching the fuel source from glycogen to triglycerides under prolonged fasting conditions. Here is the detailed experimental data. The vagotomized mice, HVX, fasted for 24 hours, lost significantly less fat, and displayed larger adipocytes than mice with a sham surgery. The same effect as that seen after the HVX was also observed after capsaicin treatment, which chemically ablates only the afferent vagus nerve composed of unmyelinated fibers. Next, we explored the mechanism by which the liver-brain adipose neurocircuitry is activated during fasting. First, the effect of the forced preloading of glycogen was tested. When glycogen synthase 2-GYS2 was overexpressed by adenovol gene delivery to the liver, the hepatic glycogen contents and consequently the adipose tissue mass increased to the levels of those seen in vagotomized mice. Next, to distinguish which of the parameters, glycogen itself or its downstream metabolites, is the key to triggering liver-brain adipose axis activation when depleted, the glycogen phosphorylase liver type gene PYGL was knocked down using shRNA. When glycogenolysis was suppressed by this RNA interference, the liver glycogen content was elevated, and this in turn led to a decrease in lipolysis in the adipose tissue. This result demonstrates that the shortage of glycogen, but not that of the downstream metabolites, is the key to triggering the neurocircuitry. In summary, glycogen shortage in the liver triggers the liver-brain adipose neural axis, promoting fatty acid and glycerol release from white adipose tissue as a response to fasting. This is another compensatory mechanism for substrate deficiency for the hepatic glucose production. Thus, glycogen was identified as a fasting signal, but sensing mechanism is also unknown. This figure shows the change in the utilization of each macronutrient during fasting. As shown here, the switch from carbohydrate to fat occurs at an early stage during fasting, and the glycogen signal in the liver-brain adipose neurocircuitry is considered to play a role in the mechanisms of this switching. SGLT2 inhibitor is a newer drug for the treatment of diabetes, and is also characterized by its weight loss effect. By selectively inhibiting SGLT2, which is responsible for glucose reabsorption in the kidney, excess glucose in the blood is excreted into the urine. SGLT2 inhibitors cause about 60 grams per day of glucose to be excreted in the urine, even in healthy subjects. This will result in a weight loss of about 2 kilograms in about 3 months. Our recent study showed that the glycogen signal which we found was implicated in the weight loss effect of SGLT2 inhibitors. As shown in the panel E of this figure, the administration of a SGLT2 inhibitor TOFO decreased the liver glycogen content. This resulted in the decrease in body weight gain, as shown in A, and the vagotomized group showed smaller responses to the SGLT2 inhibitor. Therefore, it was shown that the liver-brain adipose circuit is involved in the mechanism of weight loss caused by SGLT2 inhibitor.